we're going to talk about association mapping, and there are some differences between association mapping and QTL mapping. So the first one, and maybe the most important, is that with association mapping we can put together a diverse group of genotypes. So we can use a diversity panel or all the breeding panel in our programs. And the idea with that is that we're gonna have in that panel or in that population all the historical recombination events and all the alleles that we have in that population. So the population is going to be more diverse, and as it's going to be more diverse, we're going to have more alleles in that population. And again, as the population is diverse, so we're going to have a lot of historical recombination events in that population. And but we have to be careful with these kind of populations, because as we are putting together a germplasm of different populations, we potentially can have a population structure in the population that we are using in the study. So as a dry bean that we have two gene pools, the Mesoamerican or Middle American and the Andean gene pool. So that we have to take care of that population structure for the analysis, because if we don't take care about the subpopulation, we are going to have problems with the identification of the regions that have association with the phenotype. So after put together all the individuals or the samples in this population to do association mapping, again we have to collect the phenotypes, and we have also to collect the genotype of those individuals. And then we're gonna try to identify the molecular markers that are associated with the phenotype. And the difference here between the association mapping and the QTR mapping is that here we have to do basically linear regressions with mixed models to identify if each marker have association with the phenotype that we are working on. And I want to compare here both approaches, so QTR mapping and association mapping. So if you remember in the previous video, we talked about the QTR analysis when we have to combine two parents that have to be contrasting in the trait that we're interested on, and then we have to generate a population. So to generate this population, we need some time and some resources in the breeding program. On the other hand, we have a diverse germplasm or a diverse population, and we don't have to generate that population, so we just can use uh, all the individuals that we have in the breeding panel or a natural population of the crop that we're working on, and then we have to collect the genotype. So as you can see here, when we combine just two parents, in the case of the QTL mapping, we are just gonna assess the effect of two alleles in the phenotype that we're interested on. But in the association mapping, we have a lot of different alleles, and I represent here the alleles with different colors. So as you can see here, we have different alleles that come from different genotypes, and that potentially can increase the use of the genomic regions that we identified with association to the phenotype. And also another important point is that, as you can see here in the diversity panel, we're going to have a lot of more recombinations than in a biparental panel. So we collect the genotype, and then we got the association analysis. And here also you can see that there are some differences, because with QTL analysis, we're going to have like some big regions in the genome that have association with the phenotype. because in biparental populations there are not a lot of recombinations, so we're going to have some like big genomic regions that have association with the phenotype. And here in the association mapping, as we have more recombinations and as the statistical approach is different, because here using again mixed models, we're going to test the association of every single marker with the phenotype. So we're going to identify like smaller regions that have association with the phenotype. And some to compare those two approaches. So again, this is really important with biparental population, that is the QTL approach. We are just gonna test two alleles in the case of dry beans that we can use pure lines. So we're gonna test two alleles in the population, but with a diversity panel or a natural population, we're gonna have a lot of alleles. And also in the genotyping section is important because as in biparental populations we have a kind of a low number of recombinations, we don't need a lot of molecular markers. I remember that if there are basically with the molecular markers we want to identify the recombinations in the population. So if we don't have a lot of recombination, we don't need a lot of markers. And association mapping, we need a lot of markers because we're gonna basically try to capture all the historical recombinations in the crop that we're working on. And again, with QTL, we use a usually interval mapping, but with mapping, association mapping, we use mixed models that take into account the population structure and the genetic relationship between the individuals. Uh, but that association is marker by marker, and that is why we have to do two different significant testing. With a association mapping, we have to do Bonferroni correction or false discovery rate that basically take into account all the molecular markers in the population. And in QTL analysis, usually we use the permutation test. And again, just to give you some other advantages of association mapping is that as we can test for, or we can follow, or we can capture more recombinations in a diverse population, so we're going to have a higher mapping resolution that you can see here. So the resolution in base pairs, and with association mapping, the resolution it's really here is low, so that is good because the genomic region is going to be lower when we identified a marker that it's associated with the phenotype. And in biparental populations like as F2s or back crosses, you can see that the segment or the genomic regions are going to be bigger, or that if you compare with association mapping. So we're going to have a big region in the genome that present association with the phenotype, and that is a problem because in that big region we're going to have a lot of genes to try then to identify which of that genes it's the one that has the association with the phenotype. And also in association mapping, we can test for more alleles. So here in this table, in this graphic, you can see that in association mapping, potentially we can test for more than 40 alleles in the same locus, but in biparental populations that come from pure lines, we just can assess for two different alleles, one that comes from the mother and another one that comes from the father. And also another important advantage is that in association mapping, as we can use all the population, the breeding population that we have in the program, so we don't need time to generate a population. So that means that we are gonna need less time. And there are some concepts that are important in association mapping. as linkage disequilibrium, a population structure, and some statistical approaches. So first we're gonna discuss about the LD, or linkage disequilibrium, and the LD is defined as the non-random association of alleles between loci, and that LD is represented in this graphic as red. So as in these red sections, the correlation between the markers is strong. So that means that there is not a random segregation of the markers that are in those blocks. So when there is a high LD, so that means that all the markers in this example, in this section, in this picture, segregate together, tend to segregate together in most of the cases. So that is going to hurt the resolution of the association mapping, because that means that one of the reasons that we use association mapping is to try to collect all the historical recombination events, but when the LD it's high, that it's not gonna have a really high resolution, and that depends also about the genomes of the crops that we're working on. So some genomes, it's normally they have a high LD that are like big blocks in the genome that tends to segregate together to the next generation. And the LD, as they do, if the crop have a high LD, and we have a big blocks that tend to segregate together to the next generation, so that means that also the LD have a relationship with the density of markers that we need in the population. So if we are working with a crop that have a high LD, we are not going to need a lot of molecular markers, because we just need markers that can or that we can use to identify the recombination events in the populations. And if you remember, in the first video, we talked a little bit about the pericentromeric region, that are the regions that are around the centromere. So here in this graphic, this is a chromosome. So the dark blue region represents the pericentromeric region. And in the reference genome that was published in 2014 of Common Bean, you can see that in the pericentromeric region, the recombination it's kind of or the LD it's higher than in the achromatic regions, or in the chromatic arms that are the light blue. So it's achromatic because the DNA is less condensed. So that is why in these regions, like there are more genes and there are more recombination than in the dark blue. And you can see here, for example, just focusing chromosome 1. So we have that one centimorgan, that is 1% of recombination in the population. It's equal to 5,210 kilobases. So that means that a kilobases is 1,000, so that is 5 million of bases. And in the same chromosome, but in the achromatic regions that are here in the light blue, the distance by a centimorgan is around 278 kilobases. So as you can see here, the LD not only depend of the crop or the species that you are working on, but also depend of the region in the chromosome that you are interested on. And as you can see here, that it's different in all the chromosomes between the same genome, within the same genome. So that concept of LD, it's really important. And one thing with the LD is that, as you can imagine, the markers that are closer tend to segregate together, and as the distance between the markers are bigger and bigger, the LD start to decrease and start to segregate randomly in the population. And this is the formula to estimate the LD. It's kind of easy if you remember the allelic frequency to estimate the allelic frequency in the population. So here we have the probability to get one genotype with the big A and big B that are here in this chromosome, and then you have to subtract the probability in that population or the allele frequency of big A, and then times the allele frequency of big B, and then you just have to divide that with the probability of all the alleles. And I don't want to reminder that formula, but the point here is that as you can see here, if we have two loci that are kind of close, the segregation between A and B is not going to be so frequent in the population. But if we have two markers, in this case A and C, that are far apart, the recombination is going to be more frequent in the population. So that basically it's the concept, and is why LD is important. And the another concept that is important in association mapping is the population structure. And here again we can use the formula to estimate the LD, because the population structure can cause a false positive result. So basically we can find, if we don't take into account the population structure, we can find that one molecular marker is in LD with a QTL, but that it's not gonna be uh, like reliable. It's just gonna be a false positive result, because we're putting together two subpopulations that or gene pools that don't share the allele frequency. So just to give you an idea about that, it's again the same example of the Mesoamerican lines and the Andean lines. So just imagine that in one locus we have an allele frequency of big A of 0.2. So that means that the allele frequency of little a is going to be equal to 0.8 in the Andean gene pool. And then we have, for the same locus, a different allele frequency in the Andean gene pool. So here we have a big A allele frequency of 0.6 and a little a of 0.4. So as you can imagine, if we put together those allele frequencies in this formula, we're gonna change the allele frequencies. We're not gonna work with the allele frequency neither Mesoamerican or Andean allele frequency. So that is gonna cause a false LD between markers or between markers and QTLs. So that is why we have to control for the population structure. And there are a lot of population structures. This one, the example, the A example, it's maybe the best, where we have a diverse population, but there are not like a strong subpopulation. There are not different groups. And then we have for example, here this part of the graphic represents that there is a relationship, a family relationship between the lines, but still we don't have any structure, population structure. And usually, which is normal, is this part here, the E graphic, that is that we have subpopulations. In this case, we have two subpopulations, and we have a kind of a mix of genotypes, and that is what we have in common bean, in dry bean. So obviously we can use that kind of populations. It's just that be aware that if you use a population with a structure, we have to take into account the structure of that population to try to avoid a false positive results. And there are some statistical approaches that we use in association mapping. The simplest approach is just use the SNP as fixed effects. plus the error, and that is the general linear model. And when we take into account the population structure and the kinship, that is the additive relationship between the genotypes, that is here, we use the population structure as fixed effects and the kinship as random effect. And here you just can see that here, for example, we have kind of two subpopulations, or kind of actually three subpopulations, and that it's something similar that what we have with the kinship, but in the kinship it's a little different because we're going to try to take into account it's how strong is the relationship between each line, and we're going to have a number for that. But here we're just going to try to identify what are the three populations or two populations that we have in the study. And of course, also we can use the F-test when basically we're interested to estimate what is the variance between the treatments over what is the variance within the treatment. So as large is F of this F ratio, we can basically think that there are also differences in the mean of the treatment, that in this case are the genotypes. And with a mixed model approach, basically we can estimate the p-value, or with the test also we can estimate the p-value. And the p-value, as strongest is the association, the p-value is going to be smaller and smaller. So that is why the way to represent the association mapping is using Manhattan plots, and those Manhattan plots use the p-values but use a transformation of those p-values just to be easy to visualize the results. So we take the minus or negative logarithm of the p-value and that transform the really small number to a positive number, and with that we can identify which molecular markers, or it's just to help us to visualize the results. And at the end I want to compare the QTL mapping and association mapping. So just for the QTL mapping we need to cross two parents. In association mapping we don't need to. We can do association mapping with multi-parental populations, but we don't need to cross. We just can use diversity panels to do the association mapping. The resolution is higher in association mapping, again, because we have more recombination events than that in the biparental populations. The number of alleles that we can test in association mapping is higher because as we have a diversity panel, a diverse panel, we're going to have more alleles than in biparental population. But the power is higher in biparental population, and that makes sense because in biparental population, you, as I mentioned in the previous video, one of the most important steps is to select two parents that are contrasting in the trait that you are interested on. So as we have to do that, we have to select the parents that are contrasting, that are different in the phenotype. The population is going to segregate for that trait. In association mapping, we have a lot of alleles, so there are some lines that are good, some lines that are bad, and most of them are going to be in the middle. So and are going to be a lot of alleles, different alleles that control the trait, or to test in the statistical approach. So as you can see here, use association mapping or use a diverse population is more complex than use a biparental population that we select the parents to basically have a difference in the phenotype in the population. So That is why the power in biparental population is higher. But again, if we can detect the regions with the association mapping, the resolution is going to be higher here. And I took this sentence from Dr. Bernardo when basically he mentioned that when the objective is to detect a rare variant, and that usually is the case, a QTL mapping is therefore preferred over association mapping. And that is because imagine that we're interested in the red allele here, and and that allele is just in 1% of your population. So the statistical power to detect that the effect of that allele that is present in just 1% of your population is going to be really low. But if we identify the genotype that have that allele, for example, it's an resistance, an allele that causes resistance to some disease, and we cross that genotype with an individual that in this case is the blue that is susceptible. So just doing that cross, we can increase the allele frequency of that uh, red allele, uh, and we can transfer or increase to 1% that was here in this diversity panel to 50% in this biparental population. So we're going to increase the power to detect the effect of that allele. So that is the message that I want you to think about. And in the next video, we're gonna compare or talk more about how we can use the results of QTL and GWAS or genome association. This is the end of this video.